Hello everyone and welcome to the new session of the lecture series, and today's topic is cell cycle and cell division. And within the cell cycle and cell division, we will be studying about the topic meiosis. And within meiosis, we will be studying about all the stages which are happening in meiosis, and then we will be studying about the significance, and later we will be discussing about some of the points of difference between mitosis and meiosis. And all the topics covered in this lecture are totally NCERT based. So let's start with a brief introduction of the term meiosis. So meiosis is also known as reductional division. Alright, and it was first discovered in sea urchin eggs by Oscar Hertwig in 1876. And later on, the term meiosis was coined by Farmer and Moore in 1905. So in meiosis, what happens? There is a reduction of the chromosome number into half. Okay, this is the point to remember here, that the chromosome number are reduced to exactly half. So if the organism was diploid, means it has two sets of chromosome number, then it will reduce to haploid number, means one set of chromosome number. Okay, so if the organism was diploid, then meiosis will convert it to haploid by reducing the number of chromosomes. Most commonly, meiosis takes place in the reproductive cell, or we can say germinal cells. We can also say that meiosis takes place generally during gametogenesis. What does gametogenesis means? It means gamete formation. Okay, so meiosis is very important in gamete formation. Gametes are haploid. Now, similar to mitosis, meiosis also consists of a phase, interphase, which precedes the actual nuclear division. So what happened in mitosis? We already know now that we had interphase, and then we had the prophase, then we had the metaphase and all, anaphase, and then we have telophase. Similarly, meiosis also have interphase first, and then we have the prophase, metaphase, anaphase, telophase. Okay, and then again we have a short interphase, and then again we have prophase 2, metaphase 2, anaphase 2, and telophase 2. Now let's study about the stages of the meiosis. So meiosis involves two sequential cycle of nuclear division and cell division. Okay, or also you can say cytokinesis. So nuclear division takes place 2 times, okay, as well as cytokinesis also takes place 2 times. So we can divide meiosis into first meiosis 1, and then we have the meiosis 2. Meiosis 1 is the actual reduction phase in which the reduction of chromosome takes place, and haploid organism or the haploid cells are produced. Meiosis 2 is similar to mitosis, and it is known as the equational division, means the chromosome number remains the same. Meiosis 1, okay, is further divided into prophase 1, metaphase 1, anaphase 1, and then we have the telophase 1. Meiosis 2, whereas, is further divided into prophase 2, metaphase 2, anaphase 2, and telophase 2. Now let's talk about the stages of meiosis in detail. First, let's talk about meiosis 1. So first stage of meiosis, we know, or we can say karyokinesis or the nuclear division, is prophase 1. So what happens in prophase? Prophase is the first meiotic division, okay, and is typically longer and and is typically longer and complex as compared to the prophase of mitosis. Okay, why it is more longer and complex than the prophase of mitosis? Because prophase is further divided into many stages. Let's talk about again prophase 1. So prophase 1 is further subdivided into leptotene, zygotene, pectine, diplotene and dikinesis. So that's why prophase 1 of meiosis 1 is complex and it is longer as compared to the prophase of mitosis. Prophase of mitosis is not divided further into substages, whereas prophase 1 of meiosis is divided into these 5 substages. Now let's talk about each of the substage of prophase 1. So first one is the leptotene. So what happens during leptotene? The size of the nucleus increases. This is the first observation. Second, what happens? Chromosomes are clearly visible here as single strand here. Okay, so the chromosome is condensing now and the chromosome is clearly visible, or you can say chromosome appear as single strand in leptotene. Another thing that is observed is the presence of chromomeres during leptotene. So chromomeres are what? They are the bead-like thickenings present on the chromosomes. Okay, these are known as chromomeres. And what are these bead-like thickenings? They are nothing but these are the coiled DNA threads which are present on the chromosomes and they have been named as chromomeres. So the second stage is the zygotene stage. Here, the point here to remember is that the homologous chromosome pair up here. The chromosome start to pair up and now they are known as the homologous chromosome. And this pairing of the homologous chromosome, is known as synapsis. Okay. So this is, I've shown here in the figure, this is pairing of homologous chromosome which is known as synapsis. And you can see here, this entire is a bivalent. This is the bivalent, means it consists of 2 chromosomes. Okay, that's why bivalent. So we can say it is bivalent because it consists of 2 homologous chromosomes. And we can also call bivalent as a tetrad. Why we say it is a tetrad? Because it consists of 4 chromatids. Okay, 2 chromatids from each chromosome. So in total we have 2 chromosomes. So we have 4 chromatids. So this is first chromatid, this is second chromatid, third chromatid, fourth chromatid. That's why tetrad. These 2 are, this one, you can see the green one are sister chromatids. These are sister chromatid. The purple ones, these are also sister chromatid, whereas these 2 are non-sister chromatid. One purple and one green. These are non-sister chromatid. Non-sister chromatid means chromatid from different chromosomes. So this entire structure is a synapsis. Then what happens is zygotene? A proteinaceous structure, or you can say a proteinaceous lattice is formed, okay, between the homologous chromosomes. And this proteinaceous structure or lattice is known as the synaptonymal complex. And what is the role of the synaptonymal complex? It connects the 2 homologous chromosomes. Okay. So this is the importance of synaptonymal complex. It holds or it connects the 2 homologous chromosomes together and later on recombination takes place between these 2 chromosomes. So next stage is the Pachytene stage, this stage is also known as the crossing over stage. Okay, why it is called crossing over stage? Because crossing over takes place in this stage, in Pachytene. Okay, so point here to remember is that first one is Pachytene is the longest phase of prophase 1. Second, what happens here? Condensation of the chromosome continues. So during prophase and all the substages of prophase, condensation of the chromosome is happening, and the chromosome will appear as condensed structures. It will start appearing as chromatids. Okay, so now what happens in Pachytene? Due to the condensation of the chromosomes appear as thick threads. Here you can see in this figure, again, this is a bivalent, okay, means it is a pair of homologous chromosome, and in Pachytene, I've already told you, crossing over takes place. So the crossing over, you can see crossing over has taken place between this chromosome and this chromosome. Crossing over always takes place between the non-sister chromatid. Okay, so crossing over will take place between this one and this. These are non-sister chromatid. Now you can see during crossover what happens, there is the exchange of the genetic material. So you can see crossing over took place between this chromatid and this chromatid, and there is the exchange of the genetic material. The red material, this one, has now transferred to this chromosome here. Okay, it has transferred to this chromosome, whereas you can see blue material here, or the genes present on this chromosome, has transferred on this chromosome. And crossing over is very important because it leads to the formation of recombinants. Okay, it is the cause of variation, and variation leads to evolution. So here, these two are recombinant chromatids because there is exchange of genetic material here. There is exchange of the genetic material. That's why they are known as recombinant chromatid. Okay, whereas this one and this chromatid, they are known as non-recombinant chromatids. Now, the enzyme responsible here for the crossing over is known as recombinase. So recombinase enzyme plays an important role during the crossover or the crossing over. And what happens? There is an appearance of the recombination nodules during Pachytene. So recombination nodules appear on the chromosome during the Pachytene stage. And what are these recombination nodules? They are nothing but these are the site at which crossing over occur between the two homologous chromosomes. So what happened? First was the formation of synapses. Okay, pairing of homologous chromosome. Then after synapses, formation of synaptonymal complex. And then we had the crossing over. Okay, then we have the crossing over, which leads to the exchange of the genetic material. Enzyme involved is recombinase, and there is formation of recombination nodules, which are nothing but sites of crossing over. Okay, here you can see crossing over is taking place here. Here, this is crossing over is taking place during the Pachytene. And then there is a formation of chiasma. But chiasma is not observed during Pachytene. It is observed during the diplotene, which is the next stage after Pachytene, which is the stage after Pachytene. So next stage is the diplotene. In diplotene, what happens? Breakage of the synaptonymal complex takes place because now crossing over has taken place. So synaptonymal complex is no more needed. So the cytonymal complex breaks, the homologous chromosome start to repel each other. Okay, they starting repelling each other. Initially they were coming together. Now after crossing over, they start repelling each other. But what happens? They remain attached. They remain attached at some points, okay, on the chromosome, and this point is known as the chiasmata. Okay, so chiasmata, how can you define chiasmata? Chiasmata is the point of attachment where crossing over has taken place. This is known as chiasmata or chiasma. These are X-shaped structures. Chiasmata is a X-shaped structure. Here in this figure, you can see formation of chiasma, or you can say chiasmata. These are the point of crossing over, where crossing over is taking place. The next stage and the last stage of prophase 1 is dikinesis. So what happens in dikinesis? Chromosome, they are still condensing. Okay, they condense further. This is the most significant point here. Terminalization of chiasma takes place during the dikinesis. Okay, so after chiasma formation, what happens? The chiasmata migrate to the end of the chromosome. And why do they migrate to end of the chromosome? Because the homologous chromosomes are repelling now. Okay, they are moving apart, and because of this, the chiasmata, it shifts to the end of the chromosome. And this process is known as terminalization of chiasma. And what happens at the end of the dikinesis, what happens? The nuclear membrane as well as the nucleolus, they disappear. Along with nuclear membrane and nucleolus, cell organelles also disappear. Next stage is the metaphase 1. And in metaphase 1, what happens? The bivalent, okay, they become arranged at the equator. Okay, you can see in this figure, they are, if this is the equatorial plate or the metaphase plate, we can say this plate, you can say is the metaphase plate or the equatorial plate. The bivalents are arranged here. Okay, this is similar to mitosis. And what happens here? The microtubules from the opposite pole, they attach to the kinetochore of the homologous chromosome. And what are kinetochore? Kinetochore are nothing but they are the structures present on the centromere. Kinetochores are structures present on the centromere on which spindle fibers attach. Okay, so the spindle fibers, they attached on the kinetochore. So if this is the chromosome, okay, these two are kinetochore. This is a kinetochore which is present on the centromere, and spindle fiber will attach here. Now next stage is the anaphase 1. In anaphase, what happens? Homologous chromosome now separate. Okay, and this process is known as disjunction. So the separation of homologous chromosome has been termed as disjunction. Now what is happening in anaphase 1? The chromosome of the homologous pair, they move to the opposite poles. Here you can see they are moving to the opposite poles. Point here to remember is that there is a separation of the chromosome here. Okay, there is separation of chromosome and not chromatid. That's why it is known as reductional division. So reduction is happening here in anaphase. Here what is happening? The chromosomes are being separated. That's why the number is reduced to half. So initially, if we had 46 chromosomes, okay, so 23 chromosome goes here, 23 chromosome goes to the opposite pair. So this was diploid 2n. Now we have haploid cells. N. So during anaphase, what happens? Each pole or each end receives either paternal or maternal chromosome. Okay. And what happens? There is no division of the centromere here. This is also a point to remember. There is no division of the centromere. Centromere don't divide. That's why only chromosome will separate. If centromere will divide, then chromatid will separate. Okay, but here we see division of, we see separation of the chromosome because centromere is not dividing. Next stage is the telophase stage. In telophase, what happens? There is regrouping of the chromosome at the poles. Okay, and what happens here? The chromosome now, it was condensing earlier. In telophase, what happens? The chromosomes, they uncoil. Okay, they start uncoiling and nuclear membrane and nucleolus, it reappear. So we can say here what is happening? Reverse of prophase is happening here. At the end of prophase, what was happening? Nuclear membrane, nucleolus, they were all disappearing here and the chromosome was condensing. But in telophase, what is seen? Uncoiling of chromosome is seen. Nuclear membrane and the nucleolus, they reappear. Now what happens at the end of telophase 1? Then we have the cytokinesis 1 here. Or in cytokinesis, what happens? There is division of the cytoplasm. Division of cytoplasm takes place, leading to the formation of 2 haploid cells. Okay. So cytokinesis 1, what is the product of cytokinesis 1? Or you can say what is the product of meiosis 1? 2 haploid cells. Okay. So haploid dyad of cells means 2 cells are formed after cytokinesis 1. Now after cytokinesis, a stage is observed which is known as the interphase 2, or we can also say it as interkinesis. Now interphase, what happens during the interphase of mitosis and the interphase 1 of meiosis? There is G1 phase, then we have S phase and the G2 phase. So what happens in the S phase? There is DNA synthesis. Okay. But what happens during the interphase 2? It is, first of all, it is a very short period of rest in which there is no synthesis of DNA. Okay. Means there is no S phase or the synthetic phase. Now, this was the end of meiosis 1. Now, let's start with meiosis 2, which is also known as the equational division. This is also known as equational division. Now, let's see why it is known as equational division. So, meiosis 2 is again divided into substages. First one is the prophase 2, second is the metaphase 2, then we have anaphase 2, and the last stage is telophase 2. Now, what happens during prophase 2? First of all, the prophase 2 is shorter than the prophase 1. It is not subdivided into the substages. And what happens during prophase 2? The chromatid again, they start coiling, they shorten and they thicken because of coiling. Okay. Or you can say condensing, they condense. The chromosome become visible along with their chromatids. So now because of the condensation of the chromosome, now the chromosomes are visible along with their chromatids. You can see here in this figure, we have chromatids here. This one is one chromatid. This is another chromatid. Both of them are sister chromatid. Alright. Now what happens here? The nuclear envelope again and the nucleolus, they disappear. And what happens here? We can see the formation of the spindle fibers. So this is what is happening during prophase 2 of meiosis 2. Other events that are happening during prophase 2 are, first one is the centrioles. They start to move to the opposite pole of the cells. Now what are centrioles? Centrioles are nothing but they are the cell organelles that are involved in organization of microtubule. They are involved in organization of microtubules, and microtubules will later form spindle fibers. So at the end of prophase 2, new spindle fibers, they start appearing. And how these spindle fibers are arranged? Okay, again, this point here is to remember that they are arranged at right angles to the spindle of meiosis 1. What does this mean? If this was meiosis 1, okay, if they were arranged like this, spindle fibers in meiosis 1, okay, this was in meiosis 1. In meiosis 2, what happens? They will be arranged at right angles. Means like this. Okay, they are arranged like this. This happens in meiosis 2. Now, the next stage is mitosis 2. What happens during mitosis 2? Again, the chromosome, they are arranged at the equator. Now, each chromosome consists of 2 chromatids. Okay, they consist of 2 chromatids which are joined by the centromere. Now, what happens? The spindle fibers, they get attached again to the kinetochore of the sister chromatid. Kinetochore are present on the centromere. Now we already know. Alright. Then what happens in the next stage, which is the anaphase 2? This point here is to remember that the centromere undergo division. Okay. So the centromere will divide. Okay. And the chromatids now separate here. Chromatid will separate and not chromosome. Okay, so there is separation of chromatid due to the division of the centromere. Okay, that's why it will lead to formation of cells with equal number of chromosome. Alright, that's why it is known as the equational division. Meiosis 2 is equational division. Here the chromatids are separated and each chromatid is now known as chromosome here. You can see here the chromatids have separated. Okay, and not the chromosome. Now the last stage is the telophase 2 stage here. So again, telophase 2 is, we can say, reverse of prophase. Opposite of prophase is happening here. The chromosomes, they again uncoil, they lengthen, okay, and they lose their identity or they become non-distinct. The spindle fiber, they disappear, okay, and the centriole will replicate. And what happens? The nuclear envelope, okay, reassembles around the haploid nucleus. Now cells are haploid here, okay, because of the reductional division that happened during meiosis 1. Okay, so nuclear envelope appears, nucleolus will appear. Alright. And what is the end product here? There are formation of 4 cells here. Okay, so this is the end product of meiosis, entire meiosis. 4 cells are formed here. Okay, initially 2 cells formed here. Okay, and then from these 2 cells, again 2 cells will form, and from this again 2 cells will form, and in total we have 4 unique haploid cells. So these cells are unique. Why they are unique? Because of the crossing over that has taken place. Crossing over leads to recombination, okay, which leads to variation. That's why we have 4 unique haploid cells which are formed at the end of meiosis. Okay. Then after telophase 2, we have cytokinesis 2, which leads to the formation of 4 daughter cells. In plants, what happens? The cytokinesis is through cell plate formation. Okay. And in animal cell, what happens? The cytokinesis takes place by the method of furrowing, means furrow formation. Okay. And ultimately, 4 unique daughter cells are formed. Okay. Initially 2n. From 2n we had n and n. And then again n haploid cells. And then from here 2 haploid cells. In total we have 4 haploid cells. Now let's talk about the significance of meiosis. The most important thing here to remember is that meiosis leads to the conservation of chromosome number. Okay. Now what happens? I've already told you that from 2n, from diploid, haploid cells are formed. Okay. And 2 haploid cells can again combine with each other to form, or it can restore the diploid state again. That's why there is a conservation of the chromosome number. From 2n, again 2n is a diploid organism is forming. How it is forming? By formation of these 2 are gametes. These 2 gametes, they combine with each other, okay, and form a diploid organism, thus restoring the chromosome number. What does meiosis do? It helps in the generation of the haploid spores and gametes. So meiosis is involved in gametogenesis, means gamete formation. Alright. And in meiosis, we already know that crossing over takes place. There is formation of recombinants. And this formation of recombinants leads to the genetic variability. Okay. So meiosis is the source of variation. And this variation gets accumulated over time and leads to evolution of a new species. Now let's talk about some of the difference between mitosis and meiosis. So let's talk about mitosis first. In mitosis, prophase was not that complex. It was short and it was not divided into substages. Whereas in meiosis, what was happening? The prophase 1 of meiosis is more complex and longer. Okay, and it is further subdivided into 5 stages: leptotene, zygotene, pectine, diplotin, and diakinesis. In mitosis, what happens? There is no recombination is seen. Okay, no recombination because there is no crossing over. But in meiosis, both recombination and crossing over is taking place, which leads to variation. And recombination and crossing over takes place during the pectine stage. Now again, in mitosis, no chiasma is seen, or you can say no chiasmata is seen. But whereas chiasmata is formed during the diplotin stage of the meiosis. And in mitosis, what happens? During metaphase, the univalent are arranged at the equator, whereas during the metaphase 1, the bivalents are arranged at the equator. Okay, so this was all about meiosis and the significance of meiosis. And this was the end of the lecture. Thank you very much.